This is Frank Matter's artistic rendition of a patient having an asthma attack. Now, this is the closest thing that I'll get to a patient because I'm a PhD. I don't see a patient like Dr. Shin. But in real life, she's going to die if we don't give her right treatment at the right time. The reason she's going to die is because her airways are constricted and she cannot get air into her lungs. Now, asthma is a common chronic disorder. It's not only prevalent in developed countries, but in many parts of the developing countries. It now affects about 300 million people worldwide. Just in South Korea, it's estimated at 4% prevalence. So it is a global public health challenge. Now, asthma annually afflicts about 20 million healthy lives lost. In the United States, 14 million adults and 6 million children are affected. That amount to 500,000 hospitalizations, which account for $3 billion in US dollars. 2 million emergency department visits per year, which also account for 500 million a year. When you put the direct and indirect costs associated with asthma, it's actually greater than HIV and TB combined. Now, asthma is an inflammatory disorder and is characterized by airway hyperresponsiveness, that is, excessive narrowing of the airways. But the mechanism behind this excessive narrowing is really not known. Now, if we look in the airway, here are the airway smooth muscle cells. So these are muscle cells. And when these cells are stimulated and contract, it narrows the airway. So, in fact, in early 1800, Henry Heisel, the English physician, wrote a treatise on asthma and treatment, and he defined the vice in asthma consists not in the production of any special irritant, but in the irritability of the part irritated. So basically, he identified that the smooth muscle cell in the airways are major effector cell for asthma or bronchospasm. So this major effector cell responsible for bronchoconstriction. But when we look at smooth muscle cell in organ-based studies, we really don't understand the mechanical phenotype of human airway smooth muscle cell from asthmatics. And earlier studies have shown hypercontractility in organ-based studies, but many others have identified normal or even hypocontractility. Now, clearly, in earlier stage, there was limitation to clinical samples. But even with available human samples, 2 studies just recently published show, basically, we really don't understand the mechanical forces, or there is no consensus on the force, and even the guessing, force is even guessing game. Now, when you freshly isolated muscle cells from the airways or culture them, few studies have shown that they shorten faster and they actually contract more. But physical force themselves are inaccessible for observation. Now, to make the force visible, we need to imagine a new experimental tools. Now, we know for a long time that cells in culture exert forces. Now, earlier study by Arbor Harris have shown that when you put a fibroblast on a silicon rubber, they form a wrinkle. By generation of this wrinkle formation, Harris proposed that cells are exerting forces upon the substrate. Now, Mika Dembo and Yui Wang then subsequently visualize these forces and actually quantify these forces. The way they do that is using an elastic block or just polyacrylate gel. You have fluorescent beads embedded in the gel, and you can measure the formation of the individual beads as the cells are contracting the gel. Now, this is a very difficult problem. Now, Jim Butler from Harvard actually simplified the math and actually provided the exact solution to the force recovery. And here is a traction map of a human airway smooth muscle cell contracted with histamine. The color scale shows the absolute magnitude of stress, and the arrows indicate both relative direction and magnitude of the stresses. So this cell is actually activated by histamine, so they're contracting, and you can imagine highest forces are at the two opposing ends of the cells, and they're contracting toward the nucleus or the center of the cell. So you can actually really imagine this cell pulling on the substrate, generating forces. Now, applying traction microscopy, we started looking at the forces of human airway smooth muscle cells isolated from different individual lung donors. 6 non-asthmatic lung donors and 6 asthmatic lung donors. And we find that we found no within difference between the projector area and traction stress between the normal and asthmatics. But when you compare the non-asthmatics and asthmatic cells are bigger and they exert more stress. So they're actually having an increased basal tone. Now, neck contractile moment is another measure derived from stress, and this is a scalar measure of contractile strength. In the asthmatic cells, isolated from different lung donors, exert more forces. And the more forces, actually, it was independent of the prolonged cultures. Even early passage cells and late passage cells, asthmatic cells are always exerting more forces. And you can also fine-tune the matrix stiffness, and we found that even at soft or stiff matrix, which is in the range of physiological range of matrix airway stiffness, asthmatic cells isolated from asthmatic lung donors actually exert more forces. So, I think this really clearly provided evidence that asthmatic cells are really strong, just like Arnold Schwarzenegger, and non-asthmatic cells are like Woody Allen. They're very weak. So there are various other methods to look at the mechanical property of single cell. Now, in our lab, we use what's called magnetic twisting cytometry. So this is very simple. We have very small particle, which is about 5 microns in diameter, and we coat these with RGD peptides. So RGD peptides recognized by the cell surface integrin receptors, and they get entangled with the actin cytoskeleton. Since these beads are magnetic, we can magnetize the bead in one direction and subsequently apply a twisting current, small twisting current, perpendicular to it. Basically, what we're doing is forcing the bead to move back and forth. As the bead moves back and forth, what happens is the cell develops stress to resist the bead motion. So we know how much we're applying, the torque, and we can actually look at the lateral bead displacement in 5 nanometers resolution. We can actually compute what's called complex modulus or material property of the cytoskeleton. So we can measure stiffness and internal friction over a wide range of frequency, multiple range of perturbations. And we can do this in high throughput fashion, looking at hundreds, 200 cells at a time. Now, in the time domain, if you give a contracting agonist, we find that stiffness increases dose dependently. If you give relaxing agonists like isoproterenol that asthmatic patients take, we find that single cell level decrease in cell stiffness. So we use the dynamic changes in cell stiffness as a measure of single cell contraction and relaxation. Now, applying this new technique, we also find that in response to histamine and methacholine, among 6 different lung donors, this is the amount of stiffness or contraction that generated in non-asthmatics. And here are 6 asthmatic cells actually showing robust increase in cell stiffness or contraction. When you apply a nested design, a mixed effect model, we found that in response to histamine and methacholine, asthmatic cells actually stiffen more, basically contracting more. Now, if you were to relax these muscles, we find that asthmatic cells also show attenuated relaxation. It's kind of like mimicking what we find in patients. So in our lab, we developed many techniques to look at, basic material property cytoskeleton or single cell level. So we have not only have Fourier transform traction microscopy to measure force, single cell force, magnetic twisting cytometry to measure material property cytoskeleton, but more recently we have developed spontaneous nanoscale tracer motion to actually look at cytoscale remodeling dynamics in a live cell and in a real time. And another technique that we're developing now is trying to mimic this more long on a chip model, where we want to look at understanding the heterotypic cell-to-cell communication that fashions asthma phenotype. Now I'm going to switch gear a little bit and try to give you a little sense of new discovery that we have made over the years. So here's a new paradigm in sensory physiology. It states that sensory receptors are found in non-sensory tissues. Sour taste receptors are found in all along the spinal cord, and they sense small changes in pH and elicit neuroactivity. Sweet taste receptors are found in the gut, and it regulates glucose transport. In 2009, Michael Welsh from Iowa actually found that bitter taste receptors are expressed in the lung, and specifically to the airway epithelial cells. Now, bitter taste receptors were originally identified in the taste bud, and they're thought to play a role as an avoidance mechanism when you inhale plant toxicants. Now, in the airway epithelial cell, through the same signal transduction mechanism, what Michael Welsh identified is that it leads to activation of G proteins, phospholipase C, generation of IP3, and increase in calcium. And they propose that this calcium causes increase in ciliary peak frequency. So you can imagine bitter compound causing ciliary peak to go up and causes a mucus clearance. Now, bitter taste receptors are G protein coupled receptors. Now, GPCRs are the master regulator of human physiology. They involve in many cellular processes and most organ functions. And greater than 50% of all the drugs out there in the market target G protein coupled receptors. In 2012, Bob Lekowitz won Nobel Prize in Chemistry for his earlier work on G protein coupled receptors. Working with one of his earlier trainees, we found that bitter taste receptors are also expressed in the lung and in human airway smooth muscle cells. And the level of some of the bitter taste receptors expression were even higher than beta-adrenergic receptor. So the patient's asthma take the inhaler at the message level and protein. And when you activate subtype-specific bitter receptor, we also found that increase in calcium, just like what we saw with taste bud and airway epithelial cells. And the amount of the increase in calcium kind of consistent with GQ coupled receptor, histamine and methacholine, that causes contraction of smooth muscle cells. And intracellular calcium was absolutely required, because if you deplete intracellular store with calcium with dasiglucagon, you don't see increasing calcium with bitter compounds. And if you inhibit any of these processes where inhibit phospholipase C, beta agonist or IP3 receptor, you find that this calcium is attenuated. So when we saw this calcium and everything that we know about calcium in muscle, we naturally thought this was a mechanism whereby in occupation setting, if you were to inhale a toxicant, that leads to bronchoconstriction. What we found paradoxically is that these bitter compounds actually relax human airway smooth muscle. And the level of their relaxation was even greater than the conventional beta agonist. And this didn't involve cyclic AMP pathways. So this was really surprising to us. Bitter compound, increasing calcium, yet it leads to relaxation. So applying the magnetic twisting cytometry at the single cell level, we try to look at whether these compounds are actually contracting and relaxing at the single cell level. And what we find that histamine causes increase in stiffness, as we saw earlier. Isoproterenol leads to decrease in stiffness or relaxing. And we found that chloroquine and saccharin, bitter compound, actually relax human airway smooth muscle cell at the single cell level. Trying to understand how calcium can lead to muscle relaxation, to put a long story short, what we found is that calcium-activated potassium channel, large conductance potassium channel was involved. And if you were to inhibit protein kinase A, cyclic AMP-dependent pathway, it's not affected. If you inhibit phospholipase C, you can ablate the relaxation, and you can also ablate the relaxation with large conductance potassium channel inhibitor. So here's a pathway that we have identified, and this is entirely new mechanism to relax human airway smooth muscle cells and leads to bronchodilation. Bitter compound acting upon G protein coupled receptors activates phospholipase C, generate IP3, that leads to binding up to the IP3 receptor and releasing calcium from the intracellular store. And we've done some high-resolution confocal microscopy to look at where the calcium was occurring and seemed to be occurring at the cell periphery. That leads to activation of calcium-activated potassium channels. Rather than depolarizing the membrane, it hyperpolarizes the membrane and causes muscle relaxation and bronchodilation. So taken together, TACE receptor agonists can be envisioned as an add-on treatment when patients fail to achieve adequate bronchodilation with conventional beta-2 agonists. Now, this is very important because approximately 50% of asthmatic patients experience a suboptimal control with conventional beta agonists. And if you do chronic exposure, it can lead to bronchial hyperreactivity, which is gain of sensitivity to spasmogens, or tachyphylaxis, loss of clinical efficacy. And in the literature, it's thought that there are homologous and heterologous desensitization of beta agonists. This is widely appreciated in G protein coupled receptor field. That is, if you chronically activate G protein coupled receptor, it leads to desensitization, downregulation of the receptor, and loss of functional effect. So we asked whether this beta-arginine receptor desensitization could be affected, have an effect on beta-TACE receptor signal transduction and relaxation pathways. So in using cell-seam culture from different donors, these are healthy donors, what we have generated is if you overnight expose the cell to albuterol, small 1 nanogram per mil, you actually subsequently, they lose their response to isoproterenol. So you can actually determine that there is a loss of efficacy with long-term treatment. But when you in this experimental condition where you generate beta-adrenergic receptor desensitization, beta-TACE receptor response is not attenuated. Actually, the signal transfer mechanism is maintained and intact and actually relaxes human airway smooth muscle cell. And there's no evidence for physiologically relevant cross-desensitization of TACE receptor-mediated airway smooth muscle relaxation from chronic beta agonist. Now we went on to use human precision cut lung slides to show that this phenomenon is in effect in ex vivo setting where if you overnight treat with albuterol, the response to isoproterenol is decreased. So basically you get beta-arginine receptor desensitization, but beta-TACE receptor response is maintained. In human lung slices, even in airways that are subject to inflammation, such as the IL-13, beta-arginine receptor response to bronchodilate the carbonyl constricted airway is diminished. But beta-TACE receptor agonist, quinine, actually was able to relax and bronchodilate the airways even under conditions where you're exposing them to inflammation. Now, for a drug to have a desirable effect in human asthmatics, beta-TACE receptor expression and function must be intact in the patient that you want to treat. And just like other G protein coupled receptors, there are multiple modes where inflammation, genetics, and epigenetic can come together to modulate the pathways of signaling. What we found actually is that when you compare asthmatics and non-asthmatics, beta taste receptor expression actually is higher in the asthmatics. And in fact, it's not diminished. So they're intact expression levels. And their response to calcium is equal to the non-asthmatics. So their signaling pathway is intact. Now, if you stimulate normal and asthmatic with bitter compound, asthmatic cells are actually equally effective in relaxing and bronchodilating. So asthmatic airway smooth muscle cells and airways subject to inflammation have intact bitter taste receptor function. Taste receptor agonists may be useful bronchodilators in asthma as an adjuvant or replacement for beta agonists. And here are members of my lab and my collaborator Stephen Liggett. He trained with Bob Lekowitz on G protein coupled receptors. Ray Panettiere from UPenn and Julian Solway from Chicago provided the human samples. And these are my funding, current funding. Thank you.